Hello, and welcome to this presentation on the polymerase chain reaction. Over the next 40 minutes, we will build PCR up from first principles, meet 18 different variants of the technique, and for each one look at exactly how the primers have to be designed. Every variant also comes with working Python code that finds suitable primers in a given DNA sequence, and with the real output of that code. Let's begin. The presentation has 4 parts. Part 1 covers the fundamentals. What a PCR reaction actually is, what happens in each cycle, what is in the tube, and the rules that decide whether a primer is good or bad. Part 2 works through 18 variants, one at a time. For each one, we ask 2 questions. What problem does it solve? And what special constraint does it place on primer design? Part 3 compares the methods and covers controls and troubleshooting. And Part 4 is the code itself, together with the results of actually running it. Part 1. Fundamentals. PCR is a method for copying a chosen region of DNA millions of times in a test tube. Kary Mullis invented it in 1983 and received the Nobel Prize in Chemistry for it 10 years later. What made it practical was Taq polymerase, an enzyme isolated from a bacterium living in hot springs, which survives the high temperatures the reaction needs. And here is the single most important idea to take away. The specificity of the reaction does not come from the enzyme. It comes from the 2 primers you choose. The primers decide what gets amplified. Everything else is just chemistry. Every PCR cycle is 3 temperature steps. First, denaturation at 94 to 98 degrees Celsius. At that temperature, the hydrogen bonds between the 2 strands break and the double helix separates. Second, annealing at 50 to 65 degrees. This is where the primers base pair with their target sites, and it is the most delicate step, the one we control through the annealing temperature. Third, extension at 72 degrees, taking roughly 1 minute per 1,000 bases. The polymerase adds nucleotides in the 5' to 3' direction, building the new strand outward from the primer's 3' hydroxyl. Typically, we run 25 to 40 cycles. Why is it called a chain reaction? Because the product of each cycle becomes the template for the next. The amount of DNA doubles every cycle. Following the equation, final amount equals starting amount times 1 plus the efficiency, raised to the number of cycles. 30 perfect cycles from a single molecule give about 1 billion copies. In practice, the curve has 3 phases: exponential, then linear, then a plateau, as reagents run out and the product starts reannealing to itself instead of to primers. And that is exactly why quantitative PCR measures during the exponential phase. The plateau carries no quantitative information at all. So what is actually in the tube? First, template DNA, which is your sample. Second, 2 primers, at 1/10 to half a micromolar each. These are short single-stranded pieces of DNA, 18 to 25 bases long. Third, the 4 nucleotides, which are the building blocks. Fourth, the polymerase enzyme. Fifth, magnesium ions, at 1.5 to 2.5 millimolar. Magnesium is an essential cofactor. Note the trap here. Too little magnesium destroys your yield, and too much produces nonspecific bands. On top of that, the nucleotides themselves bind magnesium, so the 2 concentrations are coupled to each other. And finally, a buffer that holds the pH and ionic strength steady. Choosing the enzyme is not a technical detail. It directly changes what you get. Taq is cheap and robust, but it has no proofreading ability and makes about 1 error per 10,000 bases. It also adds a single adenine overhang, which is exploited for TA cloning. Enzymes like PFU, fusion, and Q5 carry a 3' to 5' exonuclease that corrects mistakes, making them 50 to 100 times more accurate. Use those whenever the product will be cloned, sequenced, or expressed as a protein. For long reactions, we use a blend of both types together. And there are hot start versions, where the enzyme is blocked until a heat activation step, which is the next variant we will discuss. So what is a primer? It is a short, synthetic, single-stranded piece of DNA. The polymerase cannot start a new chain from nothing. It can only extend an existing 3' end. The primer is what supplies that end. The forward primer has the same sequence as the plus strand and sits upstream of the target. The reverse primer is the reverse complement of the plus strand and sits downstream. Note carefully, both are always written 5' to 3'. That is the single most common student error. The region between the 2 primers, including them, is called the amplicon, and it is the product of the reaction. These are the 8 rules that govern every primer design, and they are what the code implements. 1. Length 18 to 25 bases. Long enough to be unique, short enough to anneal quickly. 2. Melting temperature between 55 and 65 degrees, and the 2 primers within 1 or 2 degrees of each other. 3. GC content between 40 and 60%. 4. A GC clamp, meaning a G or C as the final 3' base for stronger anchoring. 5. But no more than 3 G or C bases in the last 5, or you get nonspecific priming. 6. No hairpins and no self-dimers. 7. No complementarity between the 3' ends of the 2 primers, or you form primer-dimer. And 8. The primer must be unique, with exactly 1 binding site. The melting temperature is the temperature at which half the primer molecules are bound to the template and half are free. There are 3 ways to calculate it. The first is the Wallace rule, 2 times the number of A and T bases, plus 4 times the number of G and C. Very simple, but only valid for oligos shorter than about 14 bases. The second uses GC percentage, which is more accurate but still blind to the order of the bases. And the third is the nearest neighbor method, published by Santalucia in 1998. It sums the enthalpy and entropy changes of all 16 possible dinucleotide steps, and then corrects for salt concentration. That is what real software uses, and that is what we implemented in the code. To see why it matters, the sequences AGG and AGAG have exactly the same GC content, but different melting temperatures. Only the nearest neighbor method can see that difference. There are 2 main ways a primer sabotages its own reaction. The first is a hairpin. The primer folds back on itself, forming a self-complementary stem with a loop. A folded primer is simply not available to bind the template. The second is dimers. A self-dimer is 2 copies of the same primer annealing to each other. A cross-dimer is the forward and reverse primers annealing to each other. And the genuinely dangerous case is when the complementarity sits at the 3' end. Why there specifically? Because only a 3' end can be extended by the polymerase. So a dimer with a free 3' end becomes a product that amplifies itself, competing for primers and nucleotides, and giving a short band of 40 to 80 bases on the gel. Our code has a separate check for exactly this case. Once we have calculated a melting temperature, we still have to decide what to set on the machine. The rule of thumb is, annealing temperature equals the melting temperature of the lower primer minus 5 degrees. Set it too low and the primers tolerate mismatches, bind in the wrong places, and give nonspecific bands. Set it too high and they do not bind at all, and there is no product. That tension between specificity and yield is exactly why methods like touchdown and gradient PCR exist. And one important note, melting temperature depends on salt and primer concentration. A TM value quoted without stating the sodium, magnesium, and primer concentrations is a meaningless number. Before we go into detail, it helps to see the whole picture. The 18 variants fall into 7 families, grouped by the problem they solve. The first family controls specificity, hot start, touchdown, and nested PCR. The second scales the reaction up, multiplex for many targets, and long range for large products. The third measures quantity, real-time PCR in 2 versions, and digital PCR. The fourth starts from RNA instead of DNA. The fifth reads genetic variation, allele-specific, methylation-specific, and random amplification. The sixth reaches into unknown sequence, inverse and degenerate PCR. And the seventh builds new DNA. Every slide from here on presents one variant and the constraint it places on the primers. Part 2. The 18 variants. Let's start with the baseline, standard PCR, also called conventional or endpoint PCR. 2 primers, 25 to 40 cycles, and the result read on an agarose gel at the end. The key phrase there is endpoint. We can see whether the target was present or not, but we cannot tell how much there was, because by the end, every reaction has reached its plateau. Typical product size is 100 to 3,000 bases. The main uses are presence-absence testing, cloning fragments, and preparing templates for sequencing. Every other variant we will see is a modification of this baseline. The second method is hot start PCR. Here is the problem it solves. While you are setting the reaction up on the bench at room temperature, the primers are already annealing in all sorts of wrong places, and the polymerase, which is already active, extends them. So before the machine has even started, you have already made non-specific products and dimers. The solution is to block the enzyme until a heat activation step at 95 degrees. That blocking is done in three ways: an antibody bound to the enzyme, a reversible chemical modification, or a physical wax barrier. The result is a dramatic reduction in dimers and non-specific product. There is a practical bonus too. You can comfortably set up many reactions at room temperature. The third method is touchdown PCR, and the idea is simple and elegant. You start with an annealing temperature above the melting temperature of the primers, and lower it by half a degree to a degree in every cycle. In the first cycles, the conditions are so stringent that only a perfect match survives. By the time the temperature has dropped low enough to permit imperfect annealing, the correct product has already built up an enormous exponential lead, and it stays ahead. This is the classic rescue when you get non-specific bands and cannot redesign your primers. Notice that here the annealing temperature is not a single number, but an entire program descent. In our code, that program is calculated automatically from the primers that were found. The fourth method is nested PCR. Here we run two reactions in sequence with two pairs of primers. In the first round, an outer pair amplifies a large region. In the second round, we take a little of that product and amplify it again with an inner pair that binds inside the first product. And here is the beauty of it. If the first round produced something nonspecific, the chance that it also contains both inner binding sites is essentially zero. So the nonspecific product simply does not carry through. Sensitivity and specificity both increase, sometimes by a factor of 100 to 1,000. There is also a semi-nested version that reuses one of the outer primers. The cost of the method is a high contamination risk because you have to open the tube from the first round. The fifth method is multiplex PCR, amplifying several targets at once in the same tube. Here the design becomes a genuinely combinatorial problem, with three constraints that must all hold simultaneously. First, every primer has to work at a single annealing temperature, which means all their melting temperatures must fall inside a very narrow window. Second, the products must be distinguishable on a gel, so they need different sizes with at least 50 to 80 bases between them. Third, and hardest, no two primers in the mixture may form a dimer. Notice the size of that problem. 4 targets means 8 primers, which is 28 pairwise combinations to check. Our code builds the full all versus all dimer matrix. The method is used in pathogen panels, forensic profiling, and screening for genetic deletions. The sixth method is long-range PCR for products of 5 to 40,000 bases. That is far beyond the practical limit of ordinary TAC, which is about 3,000. Why does TAC fail at those lengths? Because when it inserts a wrong base, it has no way to correct it, and that error stalls its progress. So we use a blend of two enzymes, TAC providing speed, and a proofreading enzyme that removes wrong bases and lets synthesis continue. The primers here are longer, 25 to 35 bases, with high melting temperatures of 65 to 70 degrees. That allows a neat trick, a two-step protocol where annealing and extension happen together at the same temperature, 68 degrees. Extension times are long, roughly 1 minute per 1,000 bases. The seventh method is real-time quantitative PCR in the SYBR green version. Here we measure fluorescence at the end of every cycle, so we obtain a quantity, not just a yes or no. SYBR green is a dye that slips between the bases of double-stranded DNA and fluoresces when bound. The advantage is that it is cheap and universal, working with any target. The disadvantage is critical. It fluoresces on any double-stranded DNA, including primer dimers. A dimer looks exactly like a real product and gives you a wrong number. The central measurement is called CQ, the cycle at which the signal crosses a threshold. More starting template means a lower CQ, and a difference of 1 cycle corresponds to roughly a 2-fold difference in amount. Because of the dimer problem, a melt curve at the end of the run is mandatory. You raise the temperature gradually from 60 to 95 degrees and watch where the signal falls. 1 sharp peak means 1 clean product. An extra peak at a lower temperature means dimers. The amplicon here is deliberately short, 70 to 150 bases, to keep efficiency near 100%. The eighth method is real-time PCR with a TACMan probe. Here we add a third oligonucleotide, called the probe, which binds between the two primers. It carries a reporter dye at its 5' end and a quencher at its 3' end. As long as the probe is intact, the quencher absorbs the reporter's light and there is no signal. During extension, the polymerase reaches the probe, and its 5' to 3' exonuclease activity chews it up. The reporter is released, physically separated from the quencher, and light is emitted. And this is the crucial point. Signal is only emitted if the probe found its specific sequence. Primer dimers are simply invisible to the instrument. That is the main advantage over SYBR green. 2 design rules for the probe are critical. First, its melting temperature must be 8 to 10 degrees higher than the primers, so that it is already bound when the polymerase arrives. Second, there must be no guanine at the 5' end, because guanine quenches the reporter dye even after cleavage. Finally, you can give each probe a different dye and quantify several targets at once. The 9th method is reverse transcription PCR, which starts from RNA rather than DNA. In the first step, reverse transcriptase makes complementary DNA, called cDNA, and a normal PCR is then run on that. The reverse transcription step can be primed 3 ways. Oligo-DT, which binds the poly-A tail of messenger RNA, random hexamers, which transcribe all RNA, or a gene-specific primer. But the central design problem is a different one entirely, contaminating genomic DNA in the sample. Genomic DNA contains the same exons, so it gets amplified too and gives a completely wrong quantitative answer. There are 2 solutions, and both are implemented in the code. The first is an exon junction spanning primer. The primer sits half on the end of one exon and half on the start of the next. That continuous sequence only exists in mature RNA after the intron has been removed, so the primer simply cannot bind genomic DNA. The second solution is an intron spanning pair, with each primer in a different exon. The product from cDNA is short, while the product from genomic DNA is much larger or does not form at all. And an important note for exams. RT-PCR means reverse transcription, not real-time. Real-time is qPCR. That confusion is extremely common. The 10th method is digital PCR, and the idea here is completely different from everything so far. Instead of running one large reaction and measuring it, the sample is divided into roughly 20,000 tiny compartments, usually droplets in an emulsion. Each droplet contains 0 target molecules, or 1, or very few. The reaction is run to completion in every droplet, and then you simply count how many droplets are positive and how many are negative. That is a binary readout, which is where the name digital comes from. The big advantage is absolute quantification, with no standard curve and no reference sample. Going from the fraction of positive droplets to a copy number uses the Poisson distribution, lambda equals minus the natural logarithm of 1 minus the positive fraction. Why is that correction needed? Because some droplets receive 2 or more molecules and are still counted as just one positive droplet. The method is far more resistant to inhibitors and can detect small differences that qPCR misses. There is also an extra design step. Before partitioning, you digest the DNA with a restriction enzyme, and that enzyme must absolutely not cut inside the amplicon. The code checks that explicitly. The 11th method is asymmetric PCR. The goal here is to produce single-stranded rather than double-stranded DNA. Why would we want that? Because for sequencing, probe hybridization, or biosensors, a single strand is far more convenient. It does not need to be opened up again. The method uses the 2 primers at very different concentrations, 50 to 1, or even 100 to 1. The limiting primer runs out after a few cycles, and from that moment only one primer is working, so the reaction switches from exponential amplification to linear accumulation of a single strand. The classical version has a problem. It is inefficient and inconsistent. The improved version is called late PCR, and it fixes this by design. You deliberately give the limiting primer a higher melting temperature. The reasoning is that as its concentration falls, its effective melting temperature falls too. So if you start high, it still works at your chosen annealing temperature. Our code explicitly searches for a pair satisfying that relationship. The 12th method is colony PCR, and it is an intensely practical, everyday lab technique. After a cloning experiment, you have a plate with hundreds of colonies, and you need to know which ones took up the right fragment. Instead of growing each colony and purifying plasmid from it, you simply touch the colony with a pipette tip and put it straight into the reaction. You need an extended initial denaturation, 5 to 10 minutes at 95 degrees, to break the bacterial wall and release the plasmid. And the clever part is where the primers sit, in the vector backbone on either side of the cloning site. If the vector is empty, you get a small product. If the insert went in, you get a larger one, the size of the insert plus the distance between the primers. So the size difference is the answer. Read directly off the gel. You can also check orientation. One primer on the vector and one inside the insert give a product only if the fragment went in the right way round. TAC is perfectly adequate here because the product is purely diagnostic and is never used downstream. The 13th method is allele-specific PCR, also known as ARMS. It detects a single base difference, a SNP or point mutation, directly in the reaction itself. The principle rests on a property of the polymerase. It can barely extend a primer whose final 3' base does not match the template. So what do? We place the primer's 3' end exactly on the SNP position and make one primer for each allele. If it matches, there is product. If it does not, there is none. The gel itself is the genotype. In practice, a single mismatch is sometimes tolerated, which gives a false positive. So ARMS deliberately adds a second mismatch at position -2 or -3 from the end. One mismatch is tolerated, two together are not. The discrimination sharpens considerably. There is also a sophisticated version called tetraprimer ARMS. 2 outer and 2 inner primers give 3 different sizes, distinguishing homozygous wild type, heterozygote, and homozygous mutant, all in one tube. And an essential note: you always need an internal control, otherwise the absence of a band could mean either a missing allele or a failed reaction. The 14th method is inverse PCR, and it contains a particularly elegant idea. The problem: we know a short piece of DNA, and we want to know what lies on either side of it, in the unknown region. Ordinary PCR cannot help, because we have nowhere to place the outer primers. We do not know the sequence there. The solution has 3 steps. First, cut the DNA with a restriction enzyme that does not cut inside the known region. Second, dilute heavily and ligate. At high dilution, each fragment closes on itself and forms a circle. Third, and this is the heart of the method, design primers that point outward from the known region, away from each other. On a straight line that sounds absurd, since they are diverging. But on a circle, 2 primers pointing outward are in fact pointing towards each other, around the other side of the circle. So the product you get is precisely the unknown DNA you wanted. The method is used to map transposon insertion sites, viral integration sites, and to walk outward from a known fragment. The 15th method is methylation-specific PCR. Here we are reading an epigenetic mark rather than a sequence. Methylation of cytosine at CpG sites is a major mechanism of gene silencing. For example, the silencing of tumor suppressor genes. The problem is that ordinary PCR erases that information completely because the polymerase copies a methylated cytosine exactly like a normal one. The solution is a pretreatment with sodium bisulfite. Bisulfite converts every unmethylated cytosine into uracil, which is subsequently read as thymine. A methylated cytosine is protected from the reaction and stays a cytosine. And that is the central idea. The epigenetic difference has now become a sequence difference, and a sequence difference is something PCR can read. We then design 2 primer pairs, an M pair for the methylated sequence, and AU pair for the unmethylated one. There are several design rules here that exist in no other method. First, the primers must cover CpG sites, and ideally have one at the 3' end, where discrimination is sharpest. Second, the primers must also contain cytosines that are not in CpG sites in the original sequence. Why? Because those always get converted, so a primer containing them will only bind DNA that has been fully converted. That is effectively an internal control proving the treatment worked. Third, after conversion, the DNA is very rich in adenine and thymine, so melting temperatures are low and the usual limits have to be relaxed. And fourth, after conversion, the 2 strands are no longer complementary to each other, so you must pick one strand and design on it. The 16th method is to generate PCR. When do we use it? When we know the protein sequence, but not the DNA sequence. That happens when you want to clone a gene from a known family, or find the equivalent gene in a new species that has not been sequenced. The difficulty is that the genetic code is degenerate. Leucine has 6 codons, serine 6, arginine 6. So a protein cannot be translated back into DNA unambiguously. What we do instead is synthesize a mixture of oligonucleotides covering all the possibilities, written using agreed ambiguity codes. R means A or G, Y means C or T, and means any base. The key measure is the degeneracy, meaning how many different sequences are in that mixture. It has to be kept low, usually up to 128 or 256, because every variant is diluted by all the others, and only a tiny fraction of the mixture actually matches the target. There are 3 ways to reduce degeneracy. First, choose the stretch of protein with the fewest codon options. Second, anchor the 3' end on methionine or tryptophan, the only 2 amino acids with a single codon each. And third, use inosine at hopeless positions, because inosine pairs with all 4 bases. We also run at a lower annealing temperature than usual, since most of the mixture is not a perfect match. The 17th method is RAPD, random amplified polymorphic DNA. It is unusual because it is the only method requiring no prior sequence knowledge at all. You use a single short primer, typically 10 bases, with a GC content of 50 to 70%. You run it at a very low annealing temperature, 34 to 37 degrees. Under those conditions, the primer binds at dozens or hundreds of places across the genome. And wherever 2 binding sites happen to face each other at an amplifiable distance, a band appears. The result is a pattern of bands, which is effectively a genetic fingerprint. If one individual has a band that another lacks, that is a polymorphism. But the method has serious drawbacks. First, these are dominant markers, so you cannot distinguish a heterozygote from a homozygote. Second, the loci are anonymous. You do not know which part of the genome was amplified. And third, and worst, reproducing the result between laboratories is problematic, because any small change in conditions changes the pattern. For that reason, the method has largely been replaced by modern approaches such as S&P markers. The 18th and final method is assembly PCR, also called overlap extension PCR, or SOing. The goal is to join 2 DNA fragments into one continuous sequence, without restriction enzymes, and without leaving a scar in the sequence. We use 4 primers. 2 are ordinary outer primers. The other 2 are special inner primers called chimeric, because they have 2 parts. The 3' part anneals to their own fragment, and the 5' end carries a tail with the sequence of the other fragment. In the first round, each fragment is amplified separately, and each product ends up with a tail complementary to the other one. In the second round, the 2 products are mixed, and a few cycles are run with no primers at all. The strands anneal to each other through the shared overlap region, and each acts as a primer to extend the other until the full joined product is formed. Only then are the outer primers added to amplify it. Notice the important design point. There are 2 different melting temperatures to satisfy here. One is the primer annealing to its own template in round 1, and the other is the melting temperature of the overlap region itself, which must be at least 60 degrees for the joining step in round 2 to work. Applications include making fusion proteins, site-directed mutagenesis, and assembling synthetic genes. Before we summarize, it is worth knowing what falls outside the definition, because this is a common exam question. LAMP is loop-mediated isothermal amplification. It uses 4 to 6 primers and works at a single constant temperature, around 65 degrees, with no thermal cycler at all. That makes it very popular for field diagnostics. RPA works at 37 to 42 degrees, also isothermal. NASPA targets RNA and is used in virology. Rolling circle amplification works on circular templates. Bridge amplification is what generates the clusters on an Illumina sequencing flow cell. That one genuinely is a PCR, but a solid phase 1, since both primers are fixed to a surface. An emulsion PCR runs the reaction inside droplets, and is the direct ancestor of digital PCR. The dividing line is simple. PCR requires thermal cycling. Isothermal methods do not. Part 3. Comparison, controls, and troubleshooting. This table summarizes all 18 methods, 1 line each. The most important column is the third one, the defining primer design constraint. If you read down that column, you see the central idea of this entire presentation. Every method is the same basic reaction with one additional constraint imposed on the primers. In multiplex, it is that they must all work together. In TACMAN, that the probe must be hotter than the primers. In RT-PCR, that the primer must cross an exon junction. In allele-specific PCR, that the last base must sit on the mutation. And in inverse PCR, that the primers must point outward. That is really the message of the whole talk. The biology of each method lives in the primer design. Controls. This is the part students tend to skip, and that is a mistake, because without controls a result has no meaning. First, the no template control, everything in the tube except the DNA. If a band appears there, you have contamination or dimers, and the entire run is invalid. Second, a positive control, a template known to work. If it fails, the reaction itself failed, and a negative result in your sample tells you nothing. Third, an internal control, a second amplicon in the same tube, proving the sample contained amplifiable DNA and no inhibitors. Fourth, unique to RT-PCR, a no reverse transcriptase control. If you get a band there, you have genomic DNA contamination, which is exactly what an exon junction primer is meant to prevent. And for quantitative PCR, you also need a standard curve across 5 orders of magnitude. Efficiency should be between 90 and 110%, with a correlation coefficient above 0.99. Troubleshooting. These are the 5 problems you will actually encounter. No product at all. Usually the annealing temperature is too high, there is too little template, or there are inhibitors in the sample such as heparin, EDTA, or residual phenol. The first thing to try is a temperature gradient. Multiple bands, annealing temperature too low, too many cycles, or a primer with more than one binding site. Raise the temperature, switch to touchdown, or redesign. A smear down the lane. Too much template, too many cycles, or excess magnesium. A small band around 40 to 80 bases. That is almost always primer dimer. Redesign the 3' ends, lower the primer concentration, and switch to hot start. And a band in the no template control, contamination. Separate your pre-PCR and post-PCR areas. Use filter tips and aliquot your reagents. Part 4, and the practical half of the work, the code. We built a Python toolkit that finds suitable primers inside a given DNA sequence, with a separate script for each of the 18 methods we have seen, and the real output of running every one of them. The structure of the toolkit. Everything is written in plain standard Python, with no external libraries whatsoever. There is nothing to install and BioPython is not required. The core file is called PCR_core, and it is shared by every script. It contains nearest neighbor melting temperature calculation with salt correction, GC content, hairpin detection, self-dimer and cross-dimer scoring with special weighting on the 3' end and binding site counting on both strands. The sequences file holds the templates, with features deliberately planted in them. Exon and intron boundaries, a CPG island, a point mutation, and a plasmid carrying a cloned insert. Then there are 18 scripts, 1 per method. And note the important line at the bottom. To run everything on your own sequence, simply save it as a FASTA file named MySequence in the project folder. Every script will detect it automatically and use it instead of the demo template. How does the code decide that one primer is better than another? In 2 stages, exactly the way real design software works. The first stage is a hard filter. Any candidate violating even one constraint is rejected immediately. We check length, the melting temperature window, the GC window, repeated bases, the GC clamp, the number of G and C bases in the last 5, hairpins, self-dimers, and additional binding sites. The second stage is a weighted score of the survivors, where a lower score means a better primer. Look closely at the weights, because they encode the biology. A dimer at the 3' end is penalized 3 times more heavily than an internal one, because only a 3' end can be extended. And an extra binding site in the genome costs 10 points, which very nearly disqualifies the candidate outright. Finally, pairs are ranked. The sum of the 2 individual scores, plus a penalty for the difference in melting temperature between them, plus a penalty for cross-dimer. Running it is very simple. Open a command window in the project folder, go into the code directory, and run the file run all. It will execute all 18 scripts in sequence, taking about 2 minutes. You can of course also run each script individually. The output is saved into the results folder, 1 text file per PCR type, plus 1 combined file containing everything. The slides that follow show, for each method, the key part of the code on the left and the actual output of running it on the right. Standard PCR. The code on the left shows the parameter block that encodes the 8 design rules and the calculation of extension time from product length. The output panel on the right shows the 3 best primer pairs found in the template, with the product size, the melting temperature of each primer, and the recommended annealing temperature. Hot Start PCR. Here the code quantifies what Hot Start actually buys you. It models the primer-dimer artifact that would form at room temperature, calculates its melting temperature, and computes a risk index for each candidate pair. The output ranks the pairs by how much they would benefit from a hot start enzyme. Touchdown PCR. This script builds the actual touchdown program. It takes the melting temperature of the primers it found and derives the descending temperature ladder from it. The output is a cycle-by-cycle table of annealing temperature, ending with the block of cycles run at the final temperature. Nested PCR. The code performs a double search. First, an outer pair for a large product, then an inner pair that must fall entirely within the boundaries of the first product. The output includes an explicit nesting verification, showing the coordinates that prove the inner primers really do lie inside the outer amplicon. Multiplex PCR. This is computationally the most demanding script. It runs a backtracking search for a combination of primer pairs that satisfies all three constraints at once. The output shows the selected 4-plex panel, the ladder of product sizes with the gaps between them, and the complete 8x8 cross-dimer matrix. Long-range PCR. Here the parameters are different, longer primers and higher melting temperatures. The output shows a product of nearly 10,000 bases, along with the two-step cycling program and the extension times that such a length demands. Quantitative PCR with SYBR green. The script searches for a very short amplicon under unusually strict dimer constraints, and then simulates the melt curve. The output shows that the real product and the potential primer dimer are well separated in temperature, which is exactly what makes the melt curve a valid specificity check. TACMAN probe design. Here the code designs the probe as well. It scans candidates inside the amplicon, rejects any that begin with guanine, and verifies that the melting temperature is 8 to 10 degrees above the primers. The output shows the ranked probe candidates and a full rule-by-rule check of the one selected. Reverse transcription PCR. The script demonstrates both strategies. It finds a primer that crosses an exon junction and then proves computationally that it gives 0 binding sites on genomic DNA while binding the messenger RNA. The output also compares the product sizes obtained from cDNA and from genomic DNA. Digital PCR. Besides searching for a very short amplicon, the script tests every restriction enzyme and classifies it as safe or unsafe, according to whether it cuts inside the amplicon. The output also demonstrates the Poisson calculation that converts the fraction of positive droplets into an absolute copy number. Asymmetric and late PCR. Here the code explicitly searches for a pair satisfying the late PCR temperature relationship, where the limiting primer is hotter than the primer in excess. The output reports the difference achieved and the recommended concentration ratio between the two primers. Colony PCR. The script designs primers in the vector backbone on either side of the cloning site and calculates the two expected product sizes, empty vector versus a correct clone. The difference between those two numbers is what you read off the gel. Allele-specific PCR. The code builds two allele-specific primers whose 3' ends sit exactly on the mutation, adds the deliberate second mismatch, and then proves the discrimination in a table testing each primer against each template. The output also gives the 3 expected band sizes for tetraprimer ARMS. Inverse PCR. The script selects a suitable restriction enzyme, simulates the digestion and self-circularization, and designs the outward-facing primers. The output shows explicitly the coordinates that prove the outward orientation and the size of the product on the recircularized template. Methylation-specific PCR. The code first simulates the bisulfite treatment on both versions, methylated and unmethylated, and shows the resulting drop in GC content. It then designs the M pair and the U pair, verifies that each primer covers CpG sites, and cross-tests each pair against the opposite template. Degenerate PCR. The script backtranslates the protein sequence into DNA using ambiguity codes, calculates the degeneracy of every possible window, and selects the best one. Note in the output that it chooses the window ending on methionine over a cheaper window with a degenerate 3' base. It then confirms that the primer hits the correct position in the sequence. RAPD. Here the code screens candidate 10-base arbitrary primers, then scans the genome for every pair of binding sites facing each other at an amplifiable distance. It also models the competition between products, since a real gel shows far fewer bands than are theoretically possible. The output is a simulated fingerprint. Assembly PCR. The script builds all 4 primers, including the 2 chimeric ones with their tails, calculates the melting temperature of the overlap regions separately from the annealing temperatures, and prints the junction sequence that gets created along with the product sizes in both rounds. To summarize, PCR is 1 reaction, 3 temperatures, 2 primers, and an exponential curve. Every variant we saw today is that same basic reaction plus 1 additional constraint, and that constraint almost always lands on the primers. The practical conclusion is that specificity is something you design, not something you hope for. Matching melting temperatures, correct chemistry at the 3' end, avoiding dimers, and uniqueness in the genome. And finally, choosing the variant is really choosing the question. Is it there? Standard or nested PCR? How much is there? Quantitative or digital PCR? Which allele? Allele-specific or methylation-specific PCR? What lies next to it? Inverse PCR. And can I build it? Assembly PCR. These are the main sources. The original papers by Mallis and Zacai from the 1980s. The nearest neighbor paper by Santalucia from 1998, on which the temperature calculation in the code is based. The Mike guidelines for reporting quantitative PCR. The Primer 3 paper on primer design. And the original papers describing the ARMS method and methylation-specific PCR. Thank you for listening. All of the code is in the project folder, ready to run on your own sequence, and the full output of every script is included with this presentation. I am happy to take questions.